Thank you very much. It's my pleasure to visit you from California Institute of Technology, home of NASA's JPL for space studies, to talk to you about what we decided as a group that is a very important area, neuroscience. And to quote from Francis Crick, a very important scientific study, and it's highly vital to man because our entire view of the universe depends on it. And to break it down a bit, indeed, our perception and action on items like climate change, war, social empathy, how we understand each other and take care of each other, and faith does rely in this volume of brain matter. And importantly, the brain can be affected by grievous disorders that to date affect up to 1 billion people worldwide. And that's why it's very important to both understand and be able to act on the brain, not only to improve human health, but also maybe to improve the planet health and the way we interact with each other. And one significant barrier in doing that, and I'm very glad Professor Boyden went through a whirlwind of powerful neurotechnologies. We have optogenetics, we have chemogenetics, we have activity sensors and actuators that we can now introduce in animal models of disease through mainly transgenic methods or viral vector-based methods. And this allows us unprecedented ability for animal experimentation where we can modulate circuits, we can read out circuits, and establish the effects on behavior. However, there's an extremely big challenge to go from the small rodent brain, which is a very powerful experimental platform for neuroscience studies today, to the large human brain. And it's not only the sheer size and heterogeneity, but it's the ability to cover the necessary volume of brain matter that underlies complex behavior, such as the ones I mentioned that might be responsible for how we interact with each other and our world. And in addition, we want to be able to target these tools, such as optogenetics, to the brain across the lifespan, from the developing brain all the way to the aging brain. This is an extremely high challenge to bypass, and it will take a lot of effort and a lot of synchronization across studies to be able to introduce these tools in a way that we do not have to rely on transgenesis, which is rather slow and sometimes impossible in larger brains, and in a way that we can target specific cell types and specific regions. And this is why today I chose to focus on one potential method to achieve just that. By understanding the blood-brain barrier and by recruiting blood-brain barrier mechanisms, we might be able to evolve towards targeted, non-invasive study and repair of nervous systems. And this is the reason why we want to understand when we look at the brain and we look at cells, what's quite striking is that each one of our brain cells is within 20 micrometers or less of a vascular supply. And if we actually account for the entire vasculature in the human brain, our 85 billion neurons are serviced by a vascular highway that's up to 650 kilometers. This is an extremely powerful, already built-in delivery network for research tools and also therapeutics. In principle, we could deliver tools or therapeutics through a very simple IV injection and have those products reach to all of the cell types and all of the brain regions of interest. However, this is not quite routinely done today because of a significant barrier in the form of the blood-brain barrier. It's a cellular layer, many cell types tightly packed, that have the role to stringently regulate arrivals from the vascular highway. And it is a very good reason we have. The blood-brain barrier protects us, but also allows selective crossing. We can have immune defenses or nutrients selectively cross the blood-brain barrier to sustain our neuronal circuits and support cells. And at the same time, the blood-brain barrier is a very good defender by blocking viruses and bacteria. These viruses and bacteria cannot routinely access the transport pathways across the blood-brain barrier. So the blood-brain barrier both protects us from disease, but it's a deterrent in terms of us being able to deliver therapies. Because in theory, if we could lock onto particular transport mechanisms, we would be able to deliver therapies such as genetic material in the form of DNA for gene therapy. And this is an emerging and exciting and growing area of therapeutic neuroscience. And in addition, by understanding the rules of entry, we could also better fight pathogens and prevent entry of evolved viruses and evolved bacteria. However, our barrier is that we do not quite understand enough or all of the potential pathways to cross the blood-brain barrier. And it's quite noteworthy that the best known mechanism is in the form of transferrin receptor, a receptor that was discovered 4 decades ago and recruited for neuroscience and BBE about a decade ago. And given the importance of this topic, it is not enough to have only a handful of receptors that we understand and recruit. And in particular, it's worth pointing out that transferrin, it is present throughout the body. It's not only at the blood-brain barrier, so when you try to deliver research tools or therapies to the bloodstream, you will have a sink in non-brain organs, and also you might have potential side effects from affecting those organs. So what do we do about it? When my group at Caltech started working on this topic about a decade ago, we just didn't have enough information about the blood-brain barrier to design mechanism-informed ways to cross the blood-brain barrier. So instead, we used an approach from chemistry and chemical engineering, and we used the methods that my colleague Frances Arnold used for directed evolution of enzymes. We used it on vectors that could cross the BBB. And we focused on the adeno-associated viral vector. It's a non-pathogenic virus. Many humans have been exposed to it, but it doesn't cause disease. And what's very powerful about this AAV is that in very young mammals, when delivered to the bloodstream, it can cross the blood-brain barrier and transduce neurons to some extent. And it is enough to have been approved as a gene therapy for spinal muscular atrophy. And because of gene therapy with AAV, these children live longer and better. However, AAV9 in its form, in its unmodified form, does not easily cross the blood-brain barrier in mature organisms. And when it does so, mainly transduces astrocytes and not neurons. So our approach was to take this parent AAV9 that's used in the clinic and in patients and create a lot of diversity through molecular engineering of the capsid shell. And the diversity can be theoretical billion of variants. Then we deliver these libraries to the bloodstream of a living rodent. And we focus on very stringent selection tools based on pre-enzyme recombination. And we focus on organs of interest, such as can we get vectors for the brain, and ideally also vectors that would be targeted from peripheral organs that could be reasons for toxicity or side effects, such as the liver. And upon doing this, we can see which ones from this very vast library actually are able to transduce selectively the tissue of interest. A decade of work evolved from the AAV9 parent to an intermediate generation, PHPEB, that's widely used in rodent experimentation now, to a more recent generation, CAPB10. And what we did was to modify the protein shell of the AAV capsid at multiple surface loops. And those loops are instrumental in how the viral vectors interact with the cell surface, including the blood-brain barrier. And a decade of work took us from an AAV9 vector on the top that poorly transduces the brain, but very effective for the liver, to an intermediate version where you see good expression in the brain, but there's still presence in the liver that for us was concerning, to a most advanced generation where we have enriched expression in the brain and we detarget the liver. These vectors can be used in a way that can be controlled in terms of what cell types and regions we affect. And what I'm showing you here are brain areas targeted non-invasively through a simple bloodstream injection. And we can reach areas of interest to pathology, such as dopaminergic cells in the midbrain for Parkinson's disease, or serotonergic cells in the dorsal raphe for psychiatric indications. It's worth noting that dorsal raphe is under the aqueduct, and that makes it a very difficult surgery if the approach is to do intracranial surgery to target those cell types. You can also target inhibitory forebrains with gene regulatory elements, and you can maybe look at applications such as epilepsy. And you can target cerebellum for motor disorders. These tools have a very powerful way to highlight to us molecular mechanism of entry into different cell types and different organs. Because these libraries don't give us just like 5 or 10 interesting vectors, they give us hundreds of very interesting and diversified solutions to bypass the blood-brain barrier. So when we zoom in and look at different organs, different brain regions, and different cell types, we can start to extract patterns. We can see correlates between what it takes to cross the blood-brain barrier and, for example, enter inhibitory cells in the forebrain. And then we can explore that knowledge from these large data sets to single out on selective, powerful methods to target neurons specifically, or astrocytes, or vasculature. And in order to do that, we are able to extract capsids that serve different applications. And what we have done over a decade is to build quite a sizable toolkit that went from the ability to transduce in the rodent all the way to the ability to cross the blood-brain barrier in non-human primates, and with better results in the human cells. These tools can reach different cell types and different brain regions, and not only in the central nervous system, but also in the peripheral nervous system of high interest for pain indications. And these tools have been distributed to the community and are now enabling many scientific applications. For example, looking in cleared tissue volumes at morphology of cells of different molecular signatures and their connectivity. And also the ability to do this not only in transgenic rodent models, but importantly to do this in non-human primates. What you are looking at here is a Brainbow type non-human primate where delivery to the bloodstream of an infant macaque was able to deliver these red-green fluorescent proteins across the blood-brain barrier in order to highlight specific morphologies and connectivities. And to appreciate the volume covered, I'm showing the marmoset and macaque walkthrough. It would be very difficult to obtain this with surgical interventions where you have spot injections that have very reduced volume coverage. So you can cover the entire non-human primate brain and, in theory, the human brain by delivering this to the vasculature. And importantly, this has the ability to deliver actuators of behavior. What you are looking at here is activating non-invasively dopaminergic cells in the midbrain area, which are the cells that are damaged in Parkinson's disorder. And you can see on the lower side that the rodent is hyperlocomoting versus the control on the top. And what's important, this doesn't require any brain surgery. You can do this non-invasively and modulate a specific cell type. These tools have been distributed broadly, and numerous plasmids and viral vectors have made it into the community and enabled a very diverse type of experimentation from collaborators and independent labs. And it's worth pointing applications such as, importantly, in vivo screens, genetic screens for disease factors in rodents, but also in non-transgenic animals. So we are starting, for example, by CRISPR screens to see what kind of pathways or molecules brain-wide might we be able to modulate to improve the outcome in disease. What's interesting, though, in addition to being able to use these vectors for different research applications, we are also able to use them to learn very interesting new biology. And this is one example. When we tested these vectors selected in C57 black 6 mice, the first column there, we saw very strong performance in this strain of mice. However, when we tested it across different laboratory inbred mouse strains, we see quite a distinct behavior, which points to us the very important learning that there is heterogeneity of transport across the blood-brain barrier, even between mouse strains, and encourages us to look at the blood-brain barrier variability across species. So not only across rodent strains, and I have to emphasize that even us in this room, by virtue of our biological variables, whether it's ancestry, disease state, age, we are bound to be much more different than the laboratory inbred strains of mice. And this is very important for gene therapy clinical trials because we want to be able to find the mechanisms that are conserved and the mechanisms that vary across potential patients for enrollment. And in order to do that, we have as a goal to understand the mechanisms that underlie this transport both across and within brains. And one factor that's already quite apparent, even within brains, there are different vascular signatures that might serve, for example, cerebellum versus frontal areas. And this will allow us a delivery that can target different regions of the brain. How can we do this? One powerful approach would be to fall back exactly on the very diverse repertoire that we've been building. And by now, it's not only our group, it's many groups in academia and importantly, groups in industry. There's thousands and thousands of very powerful AAVs that have been evolved through directed evolution and have as a capability the ability to lock onto different yet unknown to us blood-brain barrier receptors. And when they do that, they actually point to us a way to just ask, how did they do it? These engineered AAVs found the best solution to cross, and all we have to do now is to display the putative receptors and apply these engineered AAVs and look for matches. And this has been a rather fruitful approach. And some surprising solutions arrived because we would expect many transporters, but one unexpected result was that an ancient conserved enzyme that goes all the way back to Rubisco pathway in bacteria, carbonic anhydrase 4, which is involved in the balance of pH in the blood. And interestingly, we also have it on our tongue to sense sparkling water. This ancient enzyme, it's highly abundant on the blood-brain barrier. So what you're looking there in purple is the abundance of this enzyme on the blood-brain barrier. And some engineered AAVs have found that to be the best way to cross the blood-brain barrier. This can be evolved to a pipeline where we can apply thousands of human membrane-associated proteins and just put engineered receptors and ask what are other solutions to cross the blood-brain barrier. And I'm sharing with you here additional kits that show more and more solutions that are not only different than the profile of transferrin on the blood-brain barrier in terms of their molecular identity, but also distinct in what organs they are expressed in, with some rather specific to the brain tissue. So now what we can do, we can take these receptors and couple the information about the structure of the AAV shell. We have a much better approach to one, engineer AAV solutions against conserved tumor receptors. So we have a high predictability of performance across species. And second, look at potential sites and modulate the affinities or the binding for particular receptors. For this goal, AlphaFold multimer has been a very powerful tool, and it allows us to not only engineer interactions de novo, but also block interactions that might not serve us well. And this is one example. We are all maybe way too familiar with the evolutions and all the variants of SARS-CoV-2 variant. And we can predict now with data science, we have the ability to take a parent pathogen and study its future molecular evolutions to maybe anticipate pandemics. Another thing we can do now, with increasing knowledge of BBB receptors, we can look might some pathogens, including SARS-CoV-2, change in a way that could dock onto one of our blood-brain barrier receptors, make it to the brain, and further increase the toll of pandemics like COVID-19. However, with knowledge of the receptors, And with anticipation from data science, we can be prepared. We can look at the interface between evolving pathogens and between receptors on the BBB, the growing knowledge of receptors, and prepare small molecule inhibitors or better solutions to block these potential future pandemics. So I hope I was able to outline why it's very important to understand receptors of the blood-brain barrier. 1 in 3 people are affected by brain disorders. And if we do have the knowledge of the blood-brain barrier, we can deliver therapeutic modalities. Nanoparticles were mentioned, ASOs, antibodies, important. Also diagnostic agents, non-invasive imaging modalities to maybe study degenerating circuits or infections, and ways to cope with this disease. And we can do this, we can extrapolate this approach not only to cross the blood-brain barrier, but to enter into specific cell types and specific regions by recruiting molecular information of the cell surface in different cell types, from astrocytes to neurons, whether they're diseased neurons or healthy neurons. And also by looking at the heterogeneity of the vasculature across the brain. Might there be different specific signatures in frontal areas versus, let's say, deep brain areas that we can recruit to deliver the therapies where they are needed? In addition, this knowledge will allow us to navigate current and potential future health threats from pathogens, and also to look at the inflammation state of the brain. And importantly, from a basic biology perspective, having this knowledge would allow us to better navigate disease state and also understand the critical biological variables. We are more alike than different, but the biological variables do make a difference in what therapy might be most effective for a certain enrolled patient. So it's very important to understand biological variability in terms of our ancestry, in terms of our life stage, or in terms of our cells. And a key power of understanding BBB biology would be the ability to now bypass brain surgery and through a simple bloodstream delivery to recruit the vascular highway to, 1, deliver therapies such as the ones that Professor Boyden talked about, multiplex tools for modulation of brain areas in a way that it's observant of the cell type complexity of the brain and the region complexity. And so we can deliver genetic tools, but it can expand beyond DNA. These receptors can be used to also deliver non-DNA modalities. And at the same time, unlocking BBB can be advantageous for therapies, but it's equally important to selectively lock these BBB portals to emerging threats that we can now anticipate through data science and structural modeling based on AlphaFold modalities. So we can tune this knowledge depending on the goal, and we can do this in a way that understanding of biological variables so we can have the biggest impact across individuals and lifespan. And I hope that this provided a background, and I will close, even in the holiday spirit, I will close through this, put you in a Christmas mode before questions. It's a very large volume. As Professor Boyden was saying, this might benefit from shrinking microscopy.